and this is a presentation of my honors thesis research. I did my research on the impact of the agricultural intensity on the microbiome of honeybee guts. So this is let's unpack that. The first thing is the microbiome of a gut is something that is a bunch of microbes, bacteria, and fungus that impact usually the health and digestion of a species. We know this in humans, there's been a lot of research on that, but it's also true of other species, and bees are one of those species. Agricultural intensity kind of refers to agricultural practice and the intensity with which agriculture is practiced. So an example might be conventional farms versus organic farms. Organic farms tend to have less interventions that impact the ecosystem that they're in, versus conventional farms, which have a little bit more use of pesticides, a little bit more monoculture, that kind of thing, which can really impact an ecosystem. I'm trying to look at how those agricultural practices impact the crops, and then those crops impact the pollinator. So a lot of research has been done on the impact of agricultural intensity on the nectar and the microbiome of the nectar of crops. And there's also been a lot of research on the impact of a microbiome of honeybees in their gut on their health. So we do know that a healthy microbiome of the gut means a healthy bee, and that agricultural intensity means a change to the diversity of the microbiome of nectar. What I did with this research is tried to connect those two findings and said, okay, look, we know that agriculture is impacting the crop, and we know that the crop impacts the bee, and we know that the microbiome of the bee impacts the health of the bee. Can we link those together and show that there's a significant influence of different practices of farming on the microbiome of the bee? And what we found was really interesting. So I'll go towards the methods first, and then we can talk about results. So what we did to do this study is we collected a bunch of honeybees from varying farms in the area. We stuck with almond farms in particular, and that was just for consistency's sake. They were all within a couple kilometers, just so we can make sure that they were all similar in their environment, but they were far enough apart that the bees couldn't have gone from one farm to the next in a normal foraging pattern. We collected about 36 bees from each farm. We had 4 farms total, 2 organic and 2 conventional, and we then dissected the bees, isolated both their crop stomach, so it's a little confusing, crop versus plant, and crop versus crop stomach, and then their intestine as well. So their stomach and their intestine were separated, and then they were ground up and processed, and the DNA within them was sequenced for bacteria. So we got the sequences of all the bacteria that were present in those parts of the gut, and then we used a couple different methods to graph those out and kind of look at the diversity within those regions of the gut. Awesome. And so what we found when we did that was that there was actually a very significant difference in the intestine of the bees between organic and conventional farms. This is interesting because the crop stomach is seen as being more variable and more easily influenced by the plant that the bees are going to. But the intestine is seen as more of a stable kind of passed down from other bees in the hive through contact of mouth, and less influenced by the immediate changes to the surrounding environment. And that's not what we saw here. We saw that there was actually a significant impact from the conventional farms of a major decrease in alpha diversity. And you can see that in the box plot on the bottom left, where the mouse is currently. And we were really interested in this, so we kind of went and we plotted them out in a PCOA, which shows the clusters are the more similar points. So anything grouped together is more similar to each other than they are to the other things. You can see the intestine and the crop kind of separately. When they're on the graph together, the intestines clump very, very closely together, and the crop is a little bit more dispersed. That was really good to see because that's very similar to what we have seen in other studies of the microbiome of the honeybee. So that was a good, kind of on the right path. And then when we separated out conventional versus organic and these things, we did actually note that there was a difference in the crop. So there wasn't a decrease in diversity in the crop stomach, but there was a change in the makeup, in the content. So those species were different between conventional and organic farms. The same is also true in intestine. There was less similarity between conventional farms and organic farms than there was within their own group. You can kind of see that from the distribution of the points if you look carefully. So those were both very significant. So what does this kind of mean? it's very interesting because what it means is that agriculture is having an influence on the bees, not just through pesticides or the things we would think of more instinctually, but from their guts as well, from their microbiome. This is really useful information because it can help inform practices and conservation for honeybees. It also is interesting in a study of community dynamics and what influences. So if we think of agriculture as being a disturbance to an environment, it's not just a disturbance on the macro level, it's also a disturbance on the micro level, and we're seeing evidence of this here. Lastly, the one part that was the most intriguing was the lack of difference in diversity in the crop, but the still present difference in diversity of the intestine. More investigation into that would be interesting. Perhaps the crop is so much more variable than the intestine that it's much more difficult to see a difference in diversity because there's such a higher level of diversity in general, whereas the intestines, perhaps that's different. This is just me spitballing. There needs to be more research to figure out why we're seeing that pattern. But it's not a pattern that's been represented in other literature, and so it is an interesting way to go forward. I'm hoping to move forward with this work with a study in strawberries, which have a larger range of practices. So rather than looking at conventional and organic specifically, we could look at more specific practices of hedgerows or monoculture versus polyculture, to kind of tease out that gradient a little bit better. I think it's also important to look at fungus. For this data, I will be sequencing the fungal data presently. So hopefully that'll give us a little bit more information as well on how that sort of side of things is being impacted. And then lastly, I'd just like to acknowledge I'm working in the Fukami lab at the Stanford Biology Department, and a lot of thanks to all of the lab members who have assisted me in helping kind of tease out my methods and helping with field collections. That's been really helpful. And to my PI, Tadashi Fukami. And then, of course, also for funding from the Stanford Major and Minor Grants and the MUIR program. Thank you so much for supporting this work. Thank you for listening.